The moderator for this session is Bill Liao, the partner of SOSV Ventures from the UK, a global investor, entrepreneur, and environmentalist. And he's going to be in conversation with two pioneers that are rewriting what it means to innovate in the age of intelligent biology. We have Ashley Zehnder, the founder of Fauna Bio, a company that is using animal genomics to unlock new therapies for human diseases. And they have been driving Fauna Bio into mainstream drug discovery with Fauna Brain and have a 494 million Eli Lilly obesity deal just now. And we have Dr. Pablo Zamora, advisor of the president of Chile and co-founder of NotCo in Chile, and that's the first Chilean unicorn valued as a 1.7 billion US dollars, and they are using AI to design a mixture of molecules to mimic the taste of animal-based foods using plant-based ingredients. So please put your hands together for Bill Liao, Ashley Zehnder, and Dr. Pablo Zamora. Welcome!
Thank you.
Great to have you.
Thank you.
Great to have you. Welcome.
Hi, thank you.
Great to have you with us.
Please. Hello. Great to see you here. We're going to jump straight in because we have a very short panel and you have two amazing people. I'm going to start with you, Ashley. When did you first realize that nature's data could transform human health?
I think it was really during our postdoc training. So I'm an exotic animal veterinarian by training. I ended up coming down to Stanford and doing a cancer biology PhD, and during my postdoc was really working at the intersection of animal health and human diseases. And my two colleagues in that postdoc, one was an evolutionary biologist, genomicist, looking at how genomes evolve over time and what that can tell us about our own biology. And one was a hibernation biologist who was studying the genes that are involved in how animals hibernate and their relationship to human disease. And we realized that the three of us together really had the insights to start to translate all the remarkable traits we can study in animals that are disease resistant and link that to human diseases. And so it was really the coming together of our three founders that lightened the spark, as you say. Yeah.
Fantastic. And Pablo, when did you realize that AI could taste?
First of all, thank you, Bill, for the question. I need to recognize that Bill and SOSV was my first investor in NotCo. Without these guys, NotCo didn't exist. So thank you for that. Applause for Bill. You know, he invested in an idea that nobody in Latin America believed at the time. So I start realizing that AI can affect taste and texture when we start observing way more strongest data and evidence in silico that we can get in the lab. So my company at the time have like a wet lab, like research on biology, cellular biology, and the speedness of AI and the kind of underlying connection on the patterns of the inputs that we put to mimic milk or meat or whatever, give us some answers that the human being, for the bias of culture, cannot eventually end up with the same solution. So we start realizing that we have novelty, novel ingredients, novel combinations, and novel process suggested by AI that me as a food tech expert couldn't bring that solution to the table. So it's kind of the serendipity of the process and understand underlying connections that the human cannot identify before.
And to both of you, like we hear AI everything, right? But there are very distinct, different flavors of AI. And, you know, we know that some AI is driven an inflection point in value.
Sure.
What are the AIs that aren't working? Like in biology, any thoughts?
Well, I think it really depends on the data and it depends on the questions that you're asking, right? I think you have to ask the right questions of the right data to make the AI useful, right? I think AI for the sake of AI or building foundation models for the sake of building foundation models is not terribly useful. So I think you really need to start with a key biological question. Are you looking for novel targets? Are you looking for novel chemistry? Are you looking to stratify patients in clinical studies? Then you need to make sure you're using the data to answer that question and use the right type of AI. So I think it's really matching that data to algorithm to biological question that makes a useful AI system, generally.
First of all, I fully agree with that. But one comment that I have is like, at least in my company and the following companies that have been using AI, is like, when the AI proposes something, the human behind the AI get that knowledge. So we can start doing better questions over time. So the accuracy of the using that data set is getting better and better, and we know which experiment need to be done to satisfy AI demands of new answers. So that is critical. So I'm not fan of AI per se. I'm fan of AI when I make the right questions, and when I have new experimental data that will support the hypothesis that I want to thread with AI. So we use like normally neural networks, like machine learning, and for all of that, we wanna use it in a rational way. The best way to demonstrate that AI was used it's really simple. It's like when we make a formulation based on AI, the best way to show to the rest is a visual. So we convert all the AI application in this company in a visual moment that the people that is not expert in AI can understand what we are doing and how we do it.
So visualization for you is
critical,
an integral and also
Absolutely.
iterative process.
Even for investors, right? When you at the time, I don't know, 5 years ago, when you was talking about AI and models, that was really abstract. ChatGPT wasn't there. Nobody was so familiar with the technology. So having visual expressions of what you are accomplished with AI was critical for the culture of the company and for understanding the relationship of technology with investment.
Yeah. And you know, large language models, when they first came out, were rejected by the scientific community because they hallucinate and they inherently hallucinate, right? There's a inherent bug in the technology. Now, of course, they're commercialized because commercially the marketing people have realized that we as humans don't care that AIs don't hallucinate.
Right.
But when you look at biology, and I'm going to throw this to you, Ashley, like what is good data? Because we're seeing in the generative world that AI is now feeding on the output of AI and without a human. So how do you maintain the purity of data? In fact, what is good data? I'll ask you, Ashley, and then I'll
So I think
flip
from our
to you,
perspective,
Pablo.
so we look naturally at how animals can resist and actually reverse disease using the same genes that you and I have, but in different ways. So for us, it's really having a very clean biological phenotype that is really well characterized in a very clean system. So for example, we can find novel targets, for example, for cardiovascular disease by sequencing 60 hibernating ground squirrels when they're actively repairing their hearts. That's data you might try to capture from 10,000 humans if you're looking at a heart GWAS or looking at genes across heart failure. And that data is very noisy in terms of figuring out what's actually protecting people from cardiac damage. So much smaller data sets, much cleaner data, and using the right algorithms. But it's really that very clean genotype to phenotype linkage and data that has really good signal to noise that you can interpret well, I think makes good data. Yeah.
Yeah, in my case, you know, we were in a space at the time that was very novel. So the first approach, because we have no money at the beginning, we scrapped all the data. We did we stole all the data possible in every single bank in the world, genome banks, you know, metabolic banks, and all the layer of complexity in the cellular level, right? Nutritional. And finally, what ended up doing is like the new data that is not present in any place was the most valuable data, because that was the only way to basically approach a way that a novel result can come in. And what we did in the past was integrate sensorial data with metagenomic data, with nutritional data, and with AI. So we start combining to develop the proxies, all sort of different dialogue, this completely different data set. Imagine that, you know, sensorial data is like graphs about smell and taste and texture. Another one, you have sequence of genomes. The next one, you have a lot of chemical compounds, and so on, so on. So the harness there was integrate the data and how we propose new set of data that is not present anywhere to make a distinguished approach.
And when you've got all this data, who owns it? Is it the scientist who discovered it? Is the company the scientist works for? Should nature itself be the actual owner of this data?
Yeah.
Like, we see this enormous concentration of wealth happening. Who owns the data?
No, that's a great question. For us, so a lot of times, if we're not collecting tissues from our own tissue collections, we're working directly with academic labs who work with emerging animal models. So one example is a spiny mouse. This is an animal that can repair damage to multiple tissues without scarring or fibrosis, including repairing things like the spinal cord and brain. There are labs around the country that are doing work with this species. We can collaborate with them, design the experiments. They ship tissues to us. We sequence it. We still own rights to work with the data. They still own rights to publish and use it for research purposes, so they get publications. We integrate it with now a very large knowledge graph that integrates our data with 35 other large databases used for drug discovery. And for now, our database is large enough that the power for us is really seeing the intersection of that data with the other data that we're mining. And so, it's really this balance of what do people need to get from the data and working with those collaborators. Do they need to sequence tissues? Do they need to publish? What do they need? And recognizing the contribution. Sometimes it's a single investigator working 20 years to optimize these models and making sure that they're seeing the value of that input. Yeah.
So in our case, public domain was a big part of it, and experimental data belong to the company, of course. Then we start seeing that the approach that we were taking was taken by other companies and other groups, and we start collecting data and collaborating with other companies to share the data. However, the novel data that is the outcome of the interaction of all the metadata sets that we use, the governance of that data was critical. Who have access to the, more than the owner, right? The owner is the company, of course, but who can get access to the data, how we manage sharing the data, and which piece of data need to be basically disclosed and which not was a critical part, because at the time we didn't have protocols to govern that data, and that was a big challenge.
Awesome. Excuse me. Ashley. Excuse me. What's the strangest, weirdest animal adaptation that might hold a drug secret?
The one that I like to whip out as an example of this is not the first time I've gotten this question. There's an animal called a scorpion mouse. What's really amazing about the scorpion mouse is when they get stung by a scorpion, they turn that venom around and turn it into a painkiller. So it actually becomes this, superpower for these scorpion mice. So it's a great example of sort of taking something that should be damaging and turning it into a positive biologically. We have a compound that's not from the scorpion mouse, but it's from the 13-line ground squirrel that's actually, advancing into IND-enabling studies that came from studying ground squirrel hearts. And this is a small molecule that we're targeting for heart failure with preserved ejection fraction, which is a kind of heart failure that increases with aging and obesity. It's about half of all heart failure cases now around the world. And so that is a program that came directly from mining ground squirrel biology.
Fantastic. And Pablo, what has a plant taught you about flavor that surprised you the most or surprised your team?
What was the question, sir, again?
What has a plant taught you about flavor that was a total surprise?
Oh, yeah. Just I was commenting specifically on that. You as human being eventually can recognize first layer of complexity of flavors, right, and texture. And we have a cultural bias saying that a garlic tastes like garlic, an onion tastes like onion, a walnut tastes like, nuts. However, if you mix it molecules in a specific physicochemical environment and you treat it correctly, you can start shifting the profile of aromatic molecules, and you can start pairing something that you never thought that could be possible. Imagine that you have a garlic with an apple with something else, that the result and the outcome doesn't taste like apple, doesn't taste like garlic, it doesn't taste like something else. It's something completely new. So for instance, when we were trying to develop a product that was very successful, it's called the nut milk, right? The flavor, the chemical flavor that gives us the animal sensation of milk is coming from pineapple and cabbage. And I was very, so why? What?
Surprising.
What the heck, right? And we find it out in a paper that a cabbage has prolactons, that is a molecule similar to the one, and with the pH and enzyme delivered by the pineapple, you have a conversion and a stabilization of a molecule that give you the sensation of milky way. So that kind of uncommon combination and process freaked me out, because at that point I was trusting more in the AI than in my own knowledge.
Wow. So this is to both of you.
Okay.
At the end of the day, metabolism is all about molecules and receptors.
Yep.
Right? And those interactions. The entire field of healthcare is somehow being treated separately from the entire field of nutrition.
Yes.
If you were to bet what's going to have more impact going forward? Brilliantly designed drugs or brilliantly designed foods?
I have my answer.
We
Basically,
probably each have
food.
our opinions, but I think it's a little bit of both, I would imagine.
Yeah, but for me,
Feel free to argue. I'm asking which is best. Come on. I'm an investor.
Food.
Am I going to back you or am I going to back you on this one?
That's a good question. I think for us, it's really what we're looking at is how humans can adapt to changes in environment, and that includes changes in diet, changes in climate, other types of changes in adaptations. So what we're learning from is how animals can massively modify their metabolism, and those are things that you can't just study in humans. And so I would say in terms of overall effect size, you're going to be able to modify an animal's basal metabolism, then you can really overcome problems with diet if you can burn that energy really quickly. And so I would say from an overall effect size perspective, being able to be more like a ground squirrel and burn up calories doesn't really matter what you're eating if it's all getting burned off. So I will still vote for metabolism.
And I mean, everyone wants a fluffy tail, right?
Right? Of course.
Yeah, in my case, I'm a militant of food as a medicine. You know, and my main character behind all of this is human gut health. If you combine establishment, I'm pretty fan of the three phases, like the prebiotic, probiotic, and postbiotic. And if we train correctly our human gut to start feeding correctly with the right nutrition, you will have all sort of physiological changes for good. So my position is like, personally, going back to the origin of food, introducing more diversifies of nutrients, try to make the link between ingredients and physiology, but train the gut to be the supporter of all that change in the diet. So food as a medicine would be my answer.
And when people hear the word food as medicine, they immediately think bitter, horrible tasting food.
Yeah, no, it's not the case.
So what does a true healthy meal actually should taste like? What should it actually taste like?
No, you can, using science and technology, you can develop like mainstream product, normal product using this kind of approach, right? You don't need to. I think that one of the mistakes that the food industry made, or the healthy food industry made, is like saying to people that what they're currently eating is incorrect. It's garbage, doesn't work, and shouldn't be eating that. That's why the spirulina happened, the curming process, you know, convincing people that they need to change their culture to adapt to healthy process is not going to happen, not even happen. So we need to use science, technology, and innovation to modify with the food industry the ingredients, the delivery, and what the penalties have to be compatible with the food for medicine concept.
So actually, last word, thank you.
Yeah.
Last word, and a few seconds left. What is it that people are still not understanding about AI and biology that's like driving you nuts?
Well, I think there's this concept in more general AI that we've hit some sort of data ceiling, right? We sort of have all the data that we need, and I think on the biology side, we're still in need of generating the kinds of quality data sets that can be used for AI and biology. So there's not, a ceiling in terms of what we need to generate for biology for AI. So there's a lot of open space in terms of novel data sets, novel perturbations, novel ways to think about that drug discovery process. We're nowhere close to hitting that kind of asymptote in terms of the data. We can't scrape the internet and get more information from our biology. We have to actually do experiments. Yeah.
So we're out of time, so thank you very much, both of you. Please have a round of applause for our wonderful guests, Ashley and Pablo.
And Bill.